Okay, hi everyone. It's Will. I'm going to be talking about behavioural genetics today. I think this is going to be broken up into several sections, and hopefully I'll try and make the sections as bite-sized as possible, so that each bit is a fairly coherent chunk. Best laid plans. Okay, so behavioural genetics. Basically, what behavioural genetics is a way in which we can try and separate out the relative contributions of genes and the environment on a number of things. I mean, many perfectly normal psychological behaviours are explored by behavioural genetics, things such as the relative contributions of genes in the environment to personality, but also things like intelligence and so on. It's probably most famously used, though, in looking at illnesses, whether they be mental illnesses such as schizophrenia, depression and so on, or whether they be physical illnesses such as chronic heart disease and cancer. So we're going to look at that, and the particular technique that we're going to be exploring today really tries to separate these two things out, although, as I will be pointing out, there are some problems with this particular process. Okay, so let's start off by discussing what genes are, because genes are fairly poorly understood by a lot of people. So let's just have a quick chat about this, just a brief summary really. When the Human Genome Project was announced, or the results of the Human Genome Project were announced in the early 2000s, a lot of people were surprised by actually how few genes humans seem to have. It was thought that we probably had about 60,000, maybe to 100,000 functional genes. And by functional genes, I mean genes that actually do something, because a lot of our stuff that's in our DNA is actually does nothing. But it turned out quite a bit lower than that, something like 20 to 25,000 genes. And this was not that much more than many other animals, and a lot of people were surprised, because given humans, intelligence and sophistication, we thought that it would need vastly more than 20 to 25,000 genes to put us together. Anyway, essentially what genes are, and we've known this really since the 1950s, are little bits of DNA. We can see here, so DNA lives in a chromosome, which we'll be meeting in a little bit. The chromosomes live in the nucleus of the cell in animals and humans, and the nucleus lives in the cell, as you can see here. So just simply strings of DNA, and basically it's read like a bunch of sentences. So going down these rungs here, these spell out the instructions on how to build various parts of the body. So these instructions can be used developmentally in order to help specify what a brain is like, or they can be read during our lives in order to manufacture digestive enzymes, neurotransmitters, new skin if we cut ourselves, new stomach cells, and so on and so forth. There's all kind of clever stuff that's going on here, and the best way of thinking about it is that it's a recipe. It's a kind of genetic recipe to actually specifying what we're like and how we behave. I need to say a little bit about where genes came from, and an important factor is a thing called mutation. So I'll briefly explain something about natural selection. I don't expect you to necessarily know a lot about this, but it just helps to contextualize what I'm going to be talking about. What I want you to think about here is imagine, looking at something like bacteria or an amoeba or something, some simple single-celled organism that reproduces simply by a process of splitting in half. So we have one bacterium, and if we observe it for long enough, pretty quickly that bacterium will divide into two, and these two organisms are usually identical to each other in terms of their genes, because the genes basically just make copies of themselves and we get exactly the same thing. So once there was one, now there's two, and those two copies are exact copies of each other. However, sometimes mistakes are made, and one of the things that I'll be discussing a little bit later are things called mutations. And a mutation is simply where that chemical, that DNA, has been changed in some way. Now, DNA is a chemical, and that can be changed by a number of things. It can be changed by the presence of other chemicals, or it can be altered by very high frequency radiation, such as X-rays. One of the reasons why X-rays are dangerous things to us, and one of the reasons why radiographers go diving behind lead-shielded windows, is because an overexposure of X-rays can lead to changes in our DNA, changes in mutations. Nuclear accidents such as that of Chernobyl of the 1980s led to lots of stillbirths, lots of deformities in babies, because the DNA of their mother's and father's sperm and eggs had actually been changed by the radiation that was present from the nuclear fallout. If you think of something like skin cancer is essentially the DNA of your skin cells being changed by high frequency radiation, in this case ultraviolet radiation. It essentially makes the genes not behave in ways that you would like them to. So mutations are relatively rare. They don't happen very often, but given the number of times that cells divide, the number of times bacteria divide, there's enough events which, even for a rare event, to happen relatively frequently over many thousands of replications. So replication just meaning two cells splitting. So most of these, as I've just said by talking about cancer and things like that, most mutations are bad things. They lead to the organism being worse than before. Very rarely, a mutation can actually be a good thing, meaning that the changed version is actually more effective at surviving, reproducing, feeding, whatever it happens to be, than the original version. And if you think of it being like this, very occasionally you might imagine copying a recipe out from a recipe book. If you make a mistake in that recipe book, most of the time it's not going to matter. You might just have misspelled the word jam or something like that. It's not very likely, is it? But occasionally you might make a mistake where you, instead of saying the correct ingredient, which is 1 teaspoon of salt, you put in a tablespoon of salt. And chances are that's going to make the meal worse than it would, rather than better. But very occasionally, a copying error might actually improve the food, very rarely. And so it is with genetic mutations. Very occasionally, the genetic mutation will actually be better than the original recipe or the original organism. If it is better, this basically means it's more likely to survive, it's more likely to have babies. So this new variant may start to outcompete the old variant and become the dominant version. So mutations are usually bad, sometimes good, and this is how natural selection works. And the process continues. The way Darwin described this is basically in 2 sentences. You can actually even distill it down to 1 if you want, but this is the Darwin's theory in 2 sentences. Evolution depends on 2 things. Heritable variation. There has to be some variation. There has to be some change in the DNA, change in the genes, otherwise natural evolution won't happen. If everything remains the same, there is no change. And this change is passed on to future generations. So if I have a particular kind of mutation in my body, then the chances are that I will pass that on to my offspring. So diseases are heritable, and positive traits are heritable too. Intelligence is heritable, extroversion is heritable, and so on. The second one is differential reproductive success. All this means is that certain organisms, by virtue of their different genes, are more likely to survive, reproduce, than other ones, and it is the successful ones, the ones that are better at surviving and reproducing, which we tend to see now, rather than the ones which were less successful. So that's evolution in a nutshell, and I'll briefly explain this using the classic story of the peppered moth, the Biston Betularia peppered moth. This is a moth which lives on tree bark, like we see here, but actually tree bark is quite often covered with another kind of planty-like thing. It's not actually a plant, it's some kind of symbiotic relationship between an algae and a fungus, but as far as we're concerned it looks a bit like a kind of mossy thing. And this is quite speckly, and you'll see this on trees, particularly in areas of low pollution, such as the Peak District. And you can see there the peppered moth is very well camouflaged against this background. Originally the peppered form was dominant. There were certain brown forms, but the brown forms were much more likely to be eaten, because you can imagine something which is dark brown over this speckled background of lichen-encrusted tree bark is going to stand out like a sore thumb and is going to be eaten by birds which prey upon these organisms. During the Industrial Revolution, the factories were pumping sulphur into the atmosphere, and this had the effect of killing the lichen, because lichen is very sensitive to pollution. And then what we then ended up with, particularly in cities, was trees that were completely lichenless. The bark was dark brown, as we saw in that original picture like that. This meant that now the peppered moth is the one which is the more conspicuous, and the brown one which is the more camouflage, the better camouflage one. So now we can see that what's called the selection pressure is reversed. Birds are now eating the peppered moths and not being able to spot the brown moths. So the peppered moths are less likely to have babies because they don't survive. The brown moths are more likely to have babies. So now we start seeing the brown moth is the one which dominates. So the brown one now competes the peppered one, but since the Clean Air Act, lichen is growing back on trees, and now we find that the peppered moth is coming back. So essentially what we've got here is two different mutant forms. We imagine a mutation caused the peppered moth to go brown periodically. Most of the time this was a negative consequences to the moth's survival. Okay, so the differential reproductive success meant the peppered one outcompeted the brown one. Once the pollution had killed the lichen and we had brown tree bark, then a mutation which produced a brown coloured moth, that now becomes the one which is less likely to be eaten. That now becomes the one that is more likely to survive, and that the brown gene is the one which tends to survive in the population. So I already mentioned mutations, random changes in DNA and therefore genes caused by toxins, radioactivity. Not that common, but amassed over many reproductive events where cells are splitting and dividing, it can happen. This is why we often find that cells which reproduce very rapidly tend to be the ones that are more susceptible to cancer, simply because there's a lot of mutation, a lot of copying going on, so more likely to make errors. If you imagine copying out a recipe a million times, you're probably more likely to make an error over those millions of times than you are if you just copied it out once. There's also other kinds of variation caused by so-called genetic swapping. I won't bother talking about flu viruses, but the most common way of forming variation is sex. This is sex. Very boring. Sex basically just describes a process of genetic mixing. This very strange picture here, I kind of like the metaphor to take from this. I think the one on the left is a sperm whale. There's a connection with sex. The thing in the middle is a fertilized egg. I have absolutely no idea how the thing on the right represents an unfertilized ovum, which is the female's contribution, but there you go. Sex is basically like shuffling cards. You imagine that the female has a hand of cards, the male has a hand of cards, and when the male and female produce their sperm and eggs, it's basically like the female shuffles her pack of cards, splits it in half, and puts one half in one of her eggs and the other half in another egg. The male does the same, so sperm, as we'll see, contain exactly half the number of genes, half the number of chromosomes, as we'll see in a second, as the adult body cells. What happens then? We've got the sperm and egg, when the fertilization is just these two half packs of cards coming together to form a full pack. Now the importance is that the female and the male are given random cards, which is why siblings are not identical to each other, because we shuffle in a pack. So the first sibling might get the ace of clubs, the two of hearts, and the three of diamonds, whereas, so not sibling, or the other sperm might get the queen, the king, and the ace. So this shuffling means that each sperm and each egg is very possibly unique, genetically speaking. Certainly with the case of eggs, which are far fewer. Females produce far fewer eggs than males produce sperm over the lifetime. But it means that every sperm is probably unique. Every egg is probably unique. So this, when these two sperm and eggs come together, it means that each sibling is going to be unique. And on average, siblings share half of their genes with each other. This is a picture of a set of chromosomes. As we saw earlier, the genes, these chromosomes are just long strands of DNA, and some of the regions of these we just say are genes. This is a human, because humans have 23 pairs of genes, and we know it's a male because we have an X chromosome, which females have and males have, but only males